Hey everybody, I'm Silas, and welcome to Psilocybology. Let's expand our mind. This is a series where we talk about the research on psilocybin, the psychedelic prodrug found in magic mushrooms. We'll be breaking down and discussing the scientific literature, one study at a time, from past to present. If you'd like to support the show, please consider becoming a patron through Patreon, where you can subscribe to give $1 or more per episode. Every bit of support helps grow the show, and I greatly appreciate it. With that, let's get started. Today's episode is on the article titled Effects of Mescaline and Psilocybin on Dominance Behavior of the Rat, published in 1967. It's also on the related article titled Hallucinogens and Dominance Behavior of the Rat, also published in 1967. Now, these papers were published by the same author who was affiliated with Stanford University that conducted the study we discussed 3 episodes ago on the inhibition of isolation-induced attack behavior, and the methods are actually fairly similar. To give just a brief recap, in case you haven't heard the episode. In that episode, the researchers raised 2 sets of mice, those that were raised in a group with other mice, and those that were raised in isolation. Then, the researchers administered one of 4 psychedelics to the mice that were raised in isolation. These drugs were either psilocybin, mescaline, LSD, or BOL-148. After administration, they then placed one group-housed mouse and one of the isolated mice into the same cage and recorded whether or not the previously isolated mouse attacked the other mouse. Normally, when mice are raised in isolation, as with most animals, they become more aggressive and more readily engage in attack behavior. After the administration of psychedelics, however, they found that the isolated mice were much less likely to attack the group-housed mice. Thus, they observed that psychedelics inhibited isolation-induced attack behavior. In addition to varying the type of drug administered, the researchers also manipulated how much time they waited after injecting the mice with the psychedelic before they placed the 2 mice together in the same cage. The aim was to see how long it took the drugs to produce the strongest outcome, i.e. the greatest reduction in attack behavior. Finally, the researchers also manipulated the dose of each of the 4 drugs, with the goal here of establishing the ED50, which stands for the dose that produces 50% of the maximal effect. Another way you can think of the ED50 is that it would make it a 50% chance that one of the isolated mice would attack one of the group-housed mice. In many ways, the methodology of today's study is similar, except this time the researchers looked at dominance behavior in rats after first restricting their food for 22 hours and then having them compete for food. First of all, I want to point out that it would have been much more valuable to study dominance behavior in humans rather than dominance behavior in rats. Not only are there a plethora of interesting ways such behavior could have been looked at, the ability to have dialogue with the participants would have provided much richer data. With rats, not only can they not give consent and they're also killed after the experiment, they also can't answer any questions we have, and thus requires us to make a lot of faulty inferences that could be riddled with bias. Given that all the drugs in this paper are incredibly safe for human consumption, which had been known at the time of this publication, there's just no justification I can find for having conducted this experiment using rat animals instead of human animals. Granted, the methodology would have likely looked very different and involved some creative solutions rather than fighting for food, but that actually would have allowed us to better define the types of dominance behavior we're interested in. We could have had participants play competitive games against each other and measured dominance behavior based on gameplay, and maybe even manipulated whether they were playing against friends versus strangers. We could have done a more covert study where we told the participants we were doing one thing, but in reality we were observing changes in dominance posturing when placed in a group setting. We could have measured related constructs such as aggression. There's literally a never-ending list of better experiments that could have been done examining the effect of psilocybin on dominance behavior, none of which would have required killing anyone. But that tangent aside, we're here to go over the research that's already been done. So let's talk about what these researchers did in a bit more detail, and then get into the results. For this experiment, rats were placed into a cage that was divided into 3 compartments. One compartment on each side of the cage to house the 2 rats, and one compartment in the middle that contained a food dispenser. After being placed in this cage, both rats were deprived of food for 22 hours. After the 22 hours, one of the two rats was randomly selected to be injected with either mescaline, psilocybin, LSD, or BOL-148. The other rat was injected with a saline solution to act as a control. I'm not sure if I've ever explicitly mentioned this in the past, but one of the reasons for using an injection of a saline solution, which is just salt water, as a control is because it's possible the act of injecting an animal might influence their behavior. For a test examining dominance, for instance, it's theoretically possible that the injection could have caused discomfort for the rat, and that discomfort could have led them to act more aggressively. In order to make sure any differences in behavior are due to the drug and not just the injection, it's good to have a control condition that also includes the injection but doesn't include the drug of interest. Anyway, after administration of the drug, the partitions keeping the two rats separated were simultaneously removed. Rats were allowed to eat for 10 minutes. Because the food dispenser in the center of the cage only allowed for one animal to eat at a time, they recorded the amount of time each rat spent eating. The rat that ended up spending the most time eating was termed the dominant rat, and the other was termed the submissive rat. As with the prior experiment we recapped at the start of the episode, the researchers of this study chose to manipulate both the quantity of each of the 4 drugs of interest and also the time after drug administration that the researchers waited to remove the partitions. They manipulated these variables in order to find out how long after administration the drugs produced the strongest effect, and also to determine the ED50, which again would be the dose that produces 50% of the maximal effect. The last part of the methods to discuss is the outcome, and I'll be honest, the researchers seem to have picked a bit of a strange outcome to me. The main outcome they chose to use was the percentage of experimental rats, meaning those who were given an active drug, that were submissive in excess of the percentage they expected to be submissive. Basically, the researchers said that they expected from prior research that by chance alone, 50% of the rats given an active drug would have been submissive. To account for this, they decided their outcome would be to take the number of submissive experimental rats minus the expected number of submissive experimental rats divided by the expected number. So for example, let's just pretend the experiment used 20 pairs of rats, so 40 in total, and one rat from each pair was given psilocybin. The experimenters would supposedly then expect by random chance alone that 10 of those experimental rats would be submissive. If after the experiment was over they ultimately only found that 10 experimental rats were in fact submissive, then it would mean psilocybin did not have any effect. If instead they found that 15 experimental rats were submissive, they'd say they found a 50% effect, because 50% more experimental rats than expected were found to be submissive. Personally, since they recorded the amount of time each rat spent in the dominance position and the so-called submissive position, I don't know why they didn't instead use as an outcome the time spent in either of those two positions. Often continuous data is much more robust than dichotomized data. The first results they go into have to do with how long the drugs take to produce the greatest effect. In these rats, they found that all 4 drugs produced their greatest effects 30 minutes post-administration. For the 0.2 milligram per kilogram dose of psilocybin, they found that 30 minutes post-administration, there were roughly 47% more submissive experimental rats than expected. Identical results were seen after a 4 milligram per kilogram dose of mescaline. And the LSD and BOL-148 produced results going in the same direction. This implies that all these drugs decreased dominance behavior, because it made them more likely to be categorized as submissive. As for examining the response to various doses in order to calculate the various ED50s, they found that the ED50s for psilocybin, mescaline, LSD, and BOL-148 were 0.215, 3.46, 0.009, and 0.75 milligrams per kilogram of each drug, respectively. What's interesting about these data are that for all 4 drugs, the ED50s are much lower for this study than the prior study on attack behavior. But we need to remember this study used rats, whereas the prior study used mice. Although personally when talking I confuse the two quite frequently, hopefully I haven't done that today, their drug metabolism is somewhat different. If you remember from the prior episode on attack behavior, I referenced a really neat study that gave a practical guide for quickly converting doses used in non-human animals to those that would be appropriate for human animals. In that episode, we discussed how the ED50 for psilocybin in mice could be converted by dividing the mouse dose by 12.3. When it comes to converting a rat dose to a human dose, however, we instead would divide that rat dose by 6.2. So that means that if 0.215 milligram per kilogram of psilocybin is effective at inhibiting dominance behavior in rats, then a dose of 0.035 milligram per kilogram would theoretically be a human equivalent dose. For a 70 kilogram or 154 pound adult, that would equal a total dose of about 2.4 milligrams of psilocybin. What's notable is that this is still much smaller than the ED50 we calculated using data from the prior study on attack behavior, which found that the adult human equivalent ED50 would be closer to 10 milligrams of psilocybin. It's possible this difference is because it's just easier to decrease dominance behavior compared to getting rid of attack behavior altogether. After all, to be labeled submissive in this study, the rat just had to not be eating from the dispenser for no more than 4 minutes and 59 seconds, just under half of the 10-minute period. This means the rat could have still exhibited some dominance behavior, just not as much as the other rat they were paired with. In contrast, the prior study measured the presence or absence of any amount of attack behavior, which I imagine would in fact require a stronger dose. This highlights how the ED50 can vary depending on the outcome of interest. Overall, the researchers claim that the 4 drugs all decrease dominance behavior in rats. As with the prior study, the same limitation exists whereby it's difficult to tell whether this decrease is because drugs like psilocybin increase prosocial types of behavior or reduces feelings of aggression, or if it's because these drugs do come with some sedating side effects and simply make animals less likely to engage in activity at all, which includes dominance behavior. If you have any thoughts on this or how you would distinguish between the two, definitely share. Anyway, that's it for today's brief episode. As always, I really hope you enjoyed the show. If you have any questions, feedback, or ideas for the podcast, please let me know. You can find out all the ways to reach me on the website, Psilocybology.org, where you can also find the full transcript for each episode if interested. Thank you all for listening, and I'll talk to you next time.